So good morning, everyone. Good morning. Good morning. Still people are joining in the class. So good morning, everyone. Yesterday, yes, we have discussed about the introduction of viruses, the general properties of viruses. And finally, we discussed about the life cycle of virus, how the life cycle of viruses will be. So there will be 7 or 8 stages will be there. The virus will attach with the host cell, then penetrate into the host cell, and nucleocapsid enter into the cytoplasm. The nucleocapsid is degraded and nucleic acid is released, and biosynthesis takes place. That means the assembly of the synthesis of viral particles and the assembly of viral particle and the structure of virus, and it will lyse the host cell and it will be released. So these things we have discussed yesterday. Okay. So now I will show a video of the lives. Please mute your mic. Please mute your mic. So I will share a video, the life cycle of virus, so that it will be useful to you.
Each step is crucial to successful replication, and replication.
Nice to see you again. We've been receiving lots of exciting questions from our viewers.
Targeting HIV replication.
So this is the virus. We have the glycoprotein, that is the spikes. This is the glycoprotein spikes.
And is therefore a potential target of antiretroviral drugs.
So this is the glycoprotein spikes. So the virus get attachment with the cell. So it move towards the cell here. So these are all the receptor in the host cell. So these are all the receptors present in our cell. So our cell has several receptor. So in that, the viral spikes, the glycoprotein of the virus. So these are the glycoprotein spikes. Through the spike, the virus gets attached to the receptor. You can see the virus get attached to the receptor. So this is the attachment, right? Attachment. And second step, after attachment, the virus penetrates into the host cell. Penetrates into the host cell slowly penetrates into the host. Actually, this is the WBC. It's a blue in color, WBC. Now the nucleocapsid is released into the host cell cytoplasm, and the nucleic acid is released. That is either DNA or RNA. Then the nucleic acid divides, and the virus starts to synthesize its particles. The virus starts its necessary particles. See here. So it uses several metabolic factors, enzymes. So the nucleic acid gets replicated here. The nucleic acid, either DNA or RNA, gets replicated, and all the other viral particles are synthesized. So the viral nucleic acid is synthesized. The viral nucleic acid is replicated inside the cell. Everything happens inside the cytoplasm. Everything happens inside our cytoplasm. So this is a step-by-step process. So biosynthesis takes place. Okay. All the viral particles are synthesized slowly. All the viral particles are synthesized slowly. And assembly takes place. All the viral particles in the assembly. So all the particles are assembled and become a virus. The nucleic acid is synthesized and it became a virus. Then at the last, it lies the cell and released. It lies the cell and the virus is released from the cell. You can see thousands of millions of viruses are releasing from the cell. Billions of viruses are releasing from the cell. So this is the life cycle of virus. I think you might have understand the life cycle of virus. I hope you might have understand the life cycle of viruses. Now, Now, today's topic, we are going to discuss about the identification of viruses under laboratory condition. How can we identify the viruses under laboratory condition? Some viruses can be observed directly in our normal laboratory microscope, right? So pox viruses, which has about 300 nanometers, sorry, it's 300 nanometer. It's wrongly given as millimeter, it is 300 micrometer, 300 microns. It's a bullet shaped virus. By staining the virus section, the virus particles can be seen under the microscope. So there are certain particles of viruses can be seen through microscope. First one is the inclusion bodies. The inclusion bodies will be oval shaped or irregular shaped bodies that may be present in the cytoplasm or in the nucleus of our cells. When there is a viral disease, like rabies, if a person has rabies infection, if there is a rabies virus infection, then these kind of inclusion bodies can be seen in our cytoplasm or in our cells. The next one is the Negri bodies. These are all intracytoplasmic, round or oval, purplish pink structure with the characteristic basophilic inner granules seen. So these Negri bodies can also be seen in rabies virus infection. When there is a rabies virus infection, we can see these kind of Negri bodies or inclusion bodies through the microscope. So once these bodies are discovered, once these bodies are observed through microscope, then we can confirm it is a rabies virus infection. Now come to the cultivation of viruses. How can we grow the viruses under laboratory condition? In the last class itself, we have clearly discussed about the viruses cannot grow in a normal laboratory media. It cannot be cultivated under inanimate media or chemical media like bacteria. So bacteria can grow in chemically artificial synthesized media or chemical synthesis media, whereas viruses require cell culture, that is living cell culture. So it cannot be grown, it cannot be cultivated under normal laboratory media. So as viruses are obligate intracellular parasites, they cannot be grown on ordinary laboratory media. It requires animate media. Animate media means cell culture media. So it cannot be grown in a normal normal chemical synthesis media. It requires a cell culture media. So what are all the cultivation methods? So there are three types of methods available for the cultivation of viruses. One is animal inoculation. Number two, embryonated egg. Number three, tissue culture. So these three methods are available for growing the viruses under laboratory condition. First one, animal inoculation. See, human volunteers were used in earlier in 1900. So when the beginning of 19th century, to cultivate the viruses, human volunteers were used. But due to high risk, because of this high risk, even You can see that the first polio vaccine was tested in, it was designed by, the first vaccine was designed by Edward Jenner. You know that the first vaccine, that is the cowpox vaccine, was discovered by Edward Jenner, and he tested the vaccine in his 14 years old son's body. You know that history? You know the story? The first vaccine which was introduced into the human society, was tested on a human volunteer, and that was the son of Edward Jenner, and he tested the vaccine on his own son's body, and that 14-year-old boy was died, and it was still written on his graveyard, still written on his cemetery, that he was the first human volunteer. So due to the high risk of using the human volunteer, then monkeys were used for polio viruses by Landsteiner and Popper in 1909. But as it was also very costly due to its high cost, mice were used for mice. You know that mice were used for isolation. So the mice were inoculated by several routes like intracerebral, subcutaneous, intraperitoneal, etc. Then other animals includes guinea pigs, rabbits were used. The growth can be indicated by either death of the animal or visible lesion, or the animal may suffer from disease. So these are all the indications by using animal inoculation. Then what is the advantage of using animal inoculation in the sense? It is used for study of pathogenesis, the immune response, epidemiology, and oncogenesis. The disadvantage includes the immunity. Sometimes the animal's immunity may interfere with the viral growth, and that the animal often harbor latent viruses. So next comes to the embryonated egg. Animate media means living media, living cell culture media. Animate, animals, living cell culture. Inanimate means non-living media. That is the chemical synthesized media. Hemant, clear? Animate media means living cell line culture or cell culture media. The cell will be live condition. Inanimate media means non-living. So chemicals are inanimate. So embryonated egg. Next is the embryonated egg. The chicken egg, embryonated chicken egg is used here. Embryonated hen's egg was first used by Good Pasture in 1931, and this method was further developed by Burnett. So the embryonated egg offers several sites for the cultivation of viruses. The embryonated egg offers several sites for cultivation of viruses. What are the several sites? So this is the embryonated egg. What are the several sites? Inoculation into chorioallantoic membrane. So you can see this is the chorioallantoic membrane. So directly we can inoculate the virus into the chorioallantoic membrane. So the virus produce visible lesions. For example, variola and vaccinia viruses are inoculated through the chorioallantoic membrane. Next, inoculation into allantoic cavity. Next, inoculation into allantoic cavity. So, this is the allantoic cavity. Which one is allantoic? So, this is the allantoic cavity. You can see this is the allantoic cavity. So, this method, so this site is used to cultivate influenza viruses, paramyxoviruses, and also for producing vaccines. Then inoculation into amniotic sac. Which one is the amniotic sac? So this is the amniotic sac. We can directly inoculate the virus into the amniotic sac of the young. So this is useful for cultivating influenza viruses. Then inoculation into directly the yolk sac. So this is the yolk sac, yellow yolk. We can directly inoculate the virus into the yolk sac, and this will be useful in the cultivation of chlamydia and rickettsial. Now next, the tissue culture. The first tissue culture in virology was used by Steinhardt and his colleagues in 1913. So he maintained the vaccinia viruses in fragments of rabbit cornea. So he just took the rabbit corneal cells and made into fragments, and he maintained the vaccinia viruses in the rabbit corneal cell. Further, please mute your mic. Please mute your mic. Participants, students, mute your mic. Then Kiran Sharma, mute your mic. So this method was further developed by Wellers and Robbins in 1949 for polioviruses. So they concluded that all the human viruses can be grown in cell culture. So they concluded that all the human viruses can be grown in cell culture. Next, come to the tissue culture. There are 3 types of tissue culture were available. So there are 3 types of tissue culture are there. So in the tissue culture, there are 3 types. Number 1, organ culture. Number 2, explant culture. Number 3, cell culture. The first one, organ culture. Here, small bits of organ can be maintained in vitro. In vitro means laboratory condition, right? Small bits of organ. See if it is a liver or lung or kidney, whatever. Small bits of the organ, small bit can be maintained in the laboratory condition for days or weeks. Who's that, Naman? So small bits of the organ can be maintained in the laboratory condition for days and weeks, and preserving their original characteristics, architecture, and function that will be used for specialized parasites of certain respiratory pathogens. So these kind of organ culture will be specially used for cultivating the respiratory viruses. So for example, tracheal ring culture. So just a small bit of the trachea is maintained, and it will be used for isolation of corona. Yes, if you want to isolate the coronaviruses are cultivated in or grown in tracheal ring organ culture. So tracheal ring organ culture. Next is the explant culture. Here, the fragment of tissue, the tissue will be minced will be that is somewhat powder like. The fragment of minced tissue can be grown as explants. The fragment of minced tissue can be grown as explants and embedded in plasma clots and also cultivated in suspensions. The best example is adenoid tissue explant culture for the isolation of adenoviruses. The third one, cell culture. Here, the cell will be washed, counted, and suspended in growth medium. That growth medium will be supplemented with the 13 essential amino acids. The cell was washed, counted, and it is suspended in a growth medium and also supplemented with 13 essential amino acids, vitamins, salts, glucose, the buffering system like bicarbonate with the 5% carbon dioxide, along with 5 to 10% cough or cough serum, and some antibiotics are added to prevent the bacterial contamination, and along with that phenol red as an indicator. See, the cell culture tubes may be incubated in a sloped horizontal position. So this will be maintained as either stationary culture or maybe rolled in a special roller drums to provide better aeration. So some fastidious viruses, that means the viruses which can grow very fast or grow only in this type of roller culture. Then the cell cultures are classified into, it was further classified into 3 types. One is the primary cell culture, second one is diploid cell culture, and third one is continuous cell culture. So these are normal cells. First one is the primary cell culture. These are normal cells, freshly taken from the body and culture. They are capable of limited growth in culture and cannot be maintained in as a serial culture. So this type of culture is used to isolate viruses and also useful in vaccine production. For example, if it is a monkey kidney cell, human embryonic cell, amnion chick embryo cell culture, these things are used as primary cell culture. Next is the diploid cell culture. So these are cells of single type of cell culture that retain the original diploid chromosome number and karyotype during serial subcultivation for limited number of times. Say, for example, a single type of cell, if it is a kidney cell that is taken and it is once again serially cultivated, serially grown, so that the same chromosome present in the cell will be repeated. The same chromosome will be repeated. Okay. Karyotypic. So it will be serial divided up to 50 times, limited number of times, about 50 serial passages, and then they undergo senescence. So this type of culture is used for isolation of fastidious pathogen and production of viral vaccine. The best example for this diploid cell culture is the human fibroblast cell culture. The next one is the continuous cell culture. So these are cells of single type. Usually, these are all obtained from the cancer cell, capable of continuous serial cultivation indefinitely. We can cultivate this particular cell culture serially and so many times. So they won't change, the character won't change. Like the example will be hepatitis 2, KB cell culture, viro cell lines, all these comes under continuous cell culture. So even though they are continuously cultured, serially cultured, and their character cannot be, the structure cannot be changed. Then, how can you identify the viral growth in this cell culture? So there are certain parameters are followed in identifying the viral growth in the cell culture. What are they? Number one, cytopathic effect, metabolic inhibition, heme absorption, interference, transformation, and immunofluorescence. So these are the certain parameters that we are following to identify whether the virus is growing in the particular cell culture. Okay. First one, we'll go to the cytopathic effect. So many viruses cause morphological changes in the cultured cell when which they grow. So whatever the cell culture, when viruses grow in the cell culture, it may cause some morphological changes in the cell culture, in the cell. So these changes can be observed by, we can easily observe these kind of changes in the microscope. And these changes are termed as cytopathic effects. And those viruses which produce these kind of cytopathic effects are called cytopathogenic viruses. Cytopathogenic viruses. The best example is the measles virus that produce syncytium formation, and similarly adenovirus produce large granular clumps resembling bunches of grapes. And second one is the metabolic inhibition. Normally in cell culture, the medium turns acid due to the cellular metabolism. Why? Because when the cell is cultured, the cell will multiply. When the cell metabolism takes place, normally the surrounding medium will be turns into acid. But when viruses grow in the cell culture, the cell metabolism is inhibited and there won't be any acid production. There will not be any acid production. So this can be made out by the color of the indicator. So when there is an indicator, that can observe by the color of the indicator incorporated in the medium. Then third one, heme absorption. When heme agglutinating viruses like influenza and para-influenza viruses are grown in the cell culture, their presence can be indicated by the addition of guinea pigs to the culture. So when the viruses are growing in the cell culture, just for example, influenza viruses and para-influenza viruses are growing in the cell culture, we can identify their growth just by adding some RBC of guinea pigs. If the viruses are multiplying in the culture, then the viruses will absorb the erythrocyte of the guinea pig onto the surface of the cell and it will produce an heme absorption. The next one, interference. The growth of non-cytopathogenic viruses in cell culture can be tested by subsequent challenge with a known cytopathogenic viruses. What happens? See, there is a non-cytopathogenic virus. It does not produce any symptoms. It does not produce cytopathogenic effect in the cell culture. At the time, we can introduce a cytopathogenic viruses into the cell culture. So what happens? The growth of the first will inhibit infection by the second virus. So in that medium, already the non-cytopathogenic viruses are there. When we introduce the cytopathogenic viruses, the non-cytopathogenic viruses will dominate the second viruses and there won't be no growth of the cytopathogenic viruses. From that we can conclude that the non-cytopathogenic viruses are growing in the cell culture. Now next is the transformation. Tumor forming viruses like induce cell transformation and loss of contact inhibition so that the growth appears in a piled up fashion producing microtubules. The final one, immunofluorescence. Cell from viruses infected cultures can be stained by fluorescent conjugated antiserum and examined under UV microscope for the presence of virus antigen will give positive result. Then assay of infectivity. So there are two assays are following. One is quantal assay and is quantitative assay. In the quantal assay, it indicates only the presence or absence of the infectious viruses. The quantal assay is carried out in animals, eggs, or in tissue culture. The end point is determined by the death of the animal or production of heme agglutination in the allantoic fluid. Whereas in case of quantitative assay, there are two parameters. One is POC assay and then the plaque assay. And these assay measure the actual number of viruses. Actual number of viruses. So POC assay means the viruses that produce pox on these choriolytic membrane will produce a POC. In case of plaque, it will produce plaque like this. The monolayer culture in a pet dish after the time of absorption, like foci, it's like a plaques. So this is the plaque assay. So that's all about viral cultivation and viral growth. So just wait, I will mark the utterance. If you have any doubt, you can ask me. If you have any doubt, kindly ask me. Okay, you can leave the session. Thank you. You can leave the session. Yes, you can leave the session. Thank you. Hope you understand the class.